Buenas tardes. Bienvenidos al Seminario Departamental del Centro de Tecnología Genómica. En esta ocasión estará a cargo el seminario a cargo del estudiante Omotayo Opemipo Oyedara, alumno del programa de doctorado en Ciencias en Biotecnología y perteneciente al Laboratorio de Biomedicina Molecular. Adelante.
Good afternoon, everybody. My name is Oyedara Omotayo. I'm a PhD student in the Laboratorio de Biomedicina Molecular under the supervision of Dr. Mario Alberto Rodriguez. I will be presenting part of my doctorate degree thesis, which is titled Characterization and Genome Analysis of Bdellovibrio strains, isolated from different environmental sources in Reynosa, Tamaulipas, Mexico. Predation is a common phenomenon in nature. It cuts across different groups of organisms. It's common in plants, it's common in higher animals, in aves, the birds, in the insecta, even in the prokaryotes. Why is predation common? Why is it so important in prokaryotes? It's because predation in prokaryotes can be applied for different biocontrol purposes. Control of pathogenic organisms in medical area, for example, the control of oral infections, wound infections, respiratory infections, intestinal infections. In the non-medical fields, predation in prokaryotes can be used to control pathogens in aquaculture, farm fishery, poultry, and for the control of phytopathogens. The predatory prokaryotes use different mechanisms to attack their prey. One of the mechanisms is known as wolf pack mechanism. In this kind of mechanism, predatory prokaryotes attack their prey in groups in order to devour their prey. Example can be found in the myxococci. The second one is the direct invasion of the cytoplasm of their prey. The predatory prokaryote attaches and directly attack the prey by entering their cytoplasm to devour or to digest their cellular materials for their own growth and survival. Another method the predatory prokaryotes use is called the epibiotic mechanism of attack. In the epibiotic mechanism of attack, the predator attaches to the surface of the host or the prey and digests the cellular component of the prey with some hydrolytic enzymes. The resulting components that they derive from it, they use it for their growth. Example of such can be found in the vampirococci, from the word vampirococci, vampire, attacking from the external surface, or Bdellovibrio exovorus. Another mechanism is that by periplasmic invasion, that is, the predator attaches to the host and penetrates the periplasm or the periplasmic phase of the prey before they launch in their attack. Example of such is in Bdellovibrio bacteriovirus. In the course of my study, my target is on this Bdellovibrio bacteriovirus and possibly Bdellovibrio exovorus. Bdellovibrio bacteriovirus are very tiny predatory bacteria. They are very small in size, and they belong to the group Delta proteobacteria. They are highly motile with single flagellum. Despite the fact that they are smaller in size compared to other predatory organisms like phage, they house a large genome size, and the large genome size of about 3.8 megabase pair, coding for over 3,000 proteins, which are mostly hydrolytic proteins they use to prey upon their prey. Bdellovibrio bacteriovirus are very ubiquitous. They can be found in different environments, in water systems, like runoffs, in marine habitats, in freshwater habitats. They can also be found in the soil and the rhizosphere of plants. And they have also been detected in the guts of healthy human beings. They have also been isolated in animals like chicken and cow. Brief history of Bdellovibrio. Bdellovibrio was first discovered in 1962 by Stope and his colleague when they were trying to isolate bacteriophage for the control of phytopathogens. Bdellovibrio bacteriovirus then was described as an obligate predator. But in 1965, Shilo and Brough isolated the strain of Bdellovibrio that is capable of growing on nutrient-rich medium in the laboratory. In 1972, the first attempt to use Bdellovibrio in the control of pathogens was done by Skeff. He tried to use Bdellovibrio to control phytopathogens in soybean. And he discovered that out of the 3 strains he was using, 1 was very, very effective. In 1979, the situation arose that comparing to the current trends of antibiotics with the increase in antibiotic resistance, could prey of Bdellovibrio actually develop resistance as well? They discovered that, yes, Bdellovibrio don't kill completely their prey, but the kind of resistance they exhibit is plastic. That means if Bdellovibrio is reintroduced into the prey, they will continue their attack. In 1999, like I said earlier, I said Bdellovibrio was cultured in the lab to grow on a nutrient-rich medium. Cotter and Thomason discovered an heat locus that is responsible for the growth of Bdellovibrio on nutrient-rich media. That is, this heat locus, wherever there is a mutation at a particular point in this heat locus, they said Bdellovibrio has the ability to grow on nutrient-rich media. The 2004 rather, the first genome of Bdellovibrio was sequenced. And it was surprising that the genome houses a lot of genes coding for protease, peptidase, other electrolytic enzymes they needed to attack their prey. Apart from that, they were only able to identify only 1 insertion sequence. This is interesting because Bdellovibrio has access to its prey genetic material. Why is it that there has not been an influx of prey material into Bdellovibrio? But in 2011, with the work of Pan et al., they reported that Bdellovibrio has actually experienced horizontal gene transfer, which is mostly ancient horizontal gene transfer. And they also reported that recent prey-derived horizontal gene transfer has occurred in Bdellovibrio. In 2012, Obley et al. Isolated a strain of Bdellovibrio from River Tiber. This Bdellovibrio has a unique characteristics in the sense that it is capable of growing simultaneously as a predator as well as an host independent on plate. So they isolated this strain and characterized it. And this also concluded that it has also experienced both ancient lateral gene transfer as well as recent lateral gene transfer. In 2013, Leber et al. Carried out the predation analysis of Bdellovibrio on gram-positive bacteria. The initial study limited the ability of Bdellovibrio to attack only gram-negative bacteria. But Leber et al. Discovered that Bdellovibrio bacteriovirus is capable of inhibiting the biofilms produced by gram-positive bacteria. Apart from that, Leber et al. Also reported that Bdellovibrio uses the epibiotic mechanism to attack its prey. As well, the abundance of Bdellovibrio in humans was also reported. In 2014, the application of Bdellovibrio, the interest in the application of Bdellovibrio grew more, and they used it successfully to control Pseudomonas thulaci in the mushroom. In 2015, the diversity in Bdellovibrio, in the genus Bdellovibrio, continued as they were trying to reclassify some groups. Initially, the freshwater Bdellovibrio or the terrestrial Bdellovibrio and the marine Bdellovibrio were grouped together. But after they discovered that there are differences between the two, so they reclassified them. And the marine group was initially classified as Bacterovorax group, genus Bacterovorax. In 2015, Kova et al. Suggested that this marine Bacterovorax could as well be regrouped as Allobacterovorax. As time passes on, 2016, 2017, the interest grew on the application of Bdellovibrio, and more adaptations and tests in animal models is going on currently. Sorry. Predator life cycle of Bdellovibrio is a complex one, containing two stages: the attack phase and the interperismathic phase. During the attack phase, the highly motile Bdellovibrio locates its prey, attached to its prey. After a short period of recognition, it attaches irreversibly to the prey and penetrates into the intraperismathic space of the prey. It secretes different enzymes to lyse the genetic material and the cellular materials of the prey. At this stage, they change the configuration of the prey into what is called Bdelloplast. This Bdelloplast, the rod shape of the gram-negative bacteria, becomes round, and Bdellovibrio can have enough space to develop, and they grow and divide by septation. After septation, they release some medullinic enzymes that lyse the ghost of the prey and release another attack phase of the Bdellovibrio, which continue the cycle. This shows the different genes at the attack phase. The proteins they release are mostly the chemotaxis protein, which they use to search for the prey. And in the Bdelloplast stage, as in the round structure, they produce different enzymes they use to attack, to lyse the cellular components of the prey. Bdellovibrio bacteriovirus can exist in two phenotypes. They can either be obligate predatory bacterium, in which they only depend on the prey for survival. And in the laboratory, how do we confirm this? Is that this kind of obligate host-dependent phenotype, obligate predatory Bdellovibrio, they grow as plaque-forming units on plates when they are co-cultured with their prey. However, Bdellovibrio also can grow as host-independent phenotype in the laboratory. Like I said earlier, the reason why they can do that is because the heat locus has a mutation. This between the BD0108 tag in the Bdellovibrio G100 reference strain and part of the BD0109. This particular gene codes for type 4 pillar system. And this region of the heat locus is what controls the activity of the type 4 pillar system. This type 4 pillar system is what Bdellovibrio uses to attach and invade the prey. So, Cotter and Thomason said, if there is mutation at this region, Bdellovibrio is capable of growing on nutrient-rich media. However, Bdellovibrio strain Tiberius was capable of growing both as host-dependent as host-dependent. And subsequent studies show that there is no need for mutation at this region before Bdellovibrio can grow on nutrient-rich medium. Applications of Bdellovibrio. Bdellovibrio has a wide range of application. In agriculture, they can be used to control phytopathogens. It has been proposed to control pathogens in aquaculture, animal husbandry, and poultry. In the environmental, the potential of Bdellovibrio for sewage purification has also been proposed. In food industry, Bdellovibrio has been described to have the ability to control foodborne pathogens or food spoilage or microorganisms or bacteria. In medicine, Bdellovibrio has been studied and they have shown that it has the ability to control oral pathogens, ocular infections, control of wound infections. And because it is part of the human gut, they are suggesting it as a possible probiotics. The main objective of my work is to isolate, characterize, and carry out whole genome analysis of Bdellovibrio strains from different environmental sources in Reynosa. Because studies on Bdellovibrio, there is paucity of information on Bdellovibrio in Mexico. My study site is in Reynosa. I isolated 4 strains from Reynosa. But at the initial stage of the experiment, it was difficult for me to isolate. So I extend my tentacles to a ranch in Rio Bravo, where I was able to isolate one of the strains more. So, in all, I was able to isolate 2 strains from sewage and 3 strains from soil. The methodology for isolation of Bdellovibrio is similar to the methodology used in the isolation of bacteriophage in the environment. Soil sample and sewage samples were collected aseptically and transferred to the laboratory. In the soil, there are different microorganisms. There is need to separate Bdellovibrio from other bacteria that are in the soil. So we use centrifugation method and filtration to separate the other bacteria that we don't need for the isolation of pure Bdellovibrio. And the method used is known as double layer agar plating technique, which I shorten as DLAP, double layer agar plating technique. The phenotypic characterization of the Bdellovibrio was basically through plaque forming, checking the ability to form plaque, microscopy, including light microscopy as well as atomic force microscopy, and checking their lytic activity. It's not easy to use biochemical methods to characterize Bdellovibrio because it requires some prey to grow. So the molecular characteristic was based on the amplification of the 16SR DNA gene as well as the host interaction locus. And the 16SR gene was sequenced and used to carry out the phylogenetic analysis. The results obtained. Like I said, in the environment, there are a lot of organisms, including bacteriophages. Now, the onus lies on us how to differentiate between plaque formed by bacteriophage and plaque formed by Bdellovibrio. Usually, plaque formed by Bdellovibrio always develop after 48 hours, unlike phage that develops 24 hours or less than that. Another characteristic is that Bdellovibrio plaque, after the first time of appearance, the plaque keeps increasing in size. These are the main two features that can suggest that we have possible Bdellovibrio. And Bdellovibrio, under light microscope, Bdellovibrio is highly motile. Highly motile. In fact, some people were suggesting that it's the fastest bacteria. But some bacteria too are fast compared to Bdellovibrio. But motility is also one of the ways to differentiate or to suggest the possible presence of Bdellovibrio. Just like we can see, the fast-moving Bdellovibrio under the light microscope. We use atomic force microscopy to get the different life cycle, different stages of life cycle of Bdellovibrio. Okay, so the first stage is the attack phase. We were able to discover the attack phase of Bdellovibrio, although the flagella is not showing very well. But we were able to detect the attack phase of Bdellovibrio. This is an image of phase contrast microscope that was published by McNally in 2016. So for purpose of comparison, that was why I put it there, and the life cycle. So this is the attack phase, and we're able to detect the attack phase as well. The next stage is this attachment phase, which is this phase, attachment phase in the life cycle, attachment phase as released by McNally, and the attachment phase observed under the atomic force microscopy. The next stage is the Bdelloplast stage. This is the Bdelloplast stage where the shape of the prey is rounded by the presence of the Bdellovibrio. So, in the under the atomic force, we were able to observe this stage too. And the lytic phase, whereby the progeny of Bdellovibrio after predation or after the multiplication were released. We're able to see something like that under the atomic force microscopy. This microscopy was done in UANL in Monterey. And this is the result showing the lytic activity of Bdellovibrio. We were able to observe that Bdellovibrio were able to lyse their prey significantly compared to the control. And this was indicated by the reduction in the optical density when Bdellovibrio was cultured with its prey. Two Bdellovibrio strains were considered here. The first two Bdellovibrio strains I isolated, the SKB and the SSB. So this shows the ability of Bdellovibrio to lyse their prey. The host used for SKB was the Klebsiella, and the host used for SSB was Salmonella. These were the original prey used for their isolation. It is important to mention that the prey range of Bdellovibrio is not specific, unlike we have in the phage. So, the different Bdellovibrio strains that are isolated attack the different prey organisms that we use differently. We observed that the SKB was able to attack only about 13 out of the 36 gram-negative and gram-positive bacterial strain used for the pre-range analysis. And its attack was only limited to the member of the Enterobacteriaceae. For the SSB, it was able to attack more prey, was able to attack 22 out of 36. And it was able to attack the group Enterobacteriaceae as well as other groups such as the Pseudomonas, the Streptomonas, the Vibrio cholerae, except Citrobacter fruendi. The two other Bdellovibrio, SKUV and SERB, these were isolated from soil and these were isolated from sewage. They were able to attack 28 out of the 36 preys that were used. And one important thing about these two is that when the pre-range analysis was done using microtiter plate, they were able to reduce the turbidity of Staphylococcus epidermidis. This is a gram-positive bacteria, and Bdellovibrio has been said to only attack gram-negative bacteria. However, when the plaque analysis was done, we were not able to see a conspicuous plaque with Staphylococcus epidermidis. All the Bdellovibrio strains were not able to attack Pseudomonas syringae, Staphylococcus aureus, Rhizobium legionnarisorium, Agrobacterium tumefaciens, and Bacillus species. The molecular characterization include the amplification of 16S rDNA gene. The 5 Bdellovibrio strains that are isolated were positive for the 16S rDNA gene, while 2 were conspicuously positive for the amplification of host interaction locus, the SSB and the SCRB, both isolated from soil. The other strains were not positive for the amplification of host interaction locus. One of the strains was isolated from sewage, but when the 16S rDNA gene was blasted in the NCBI, we discovered that it grouped with the genus Bacterovorax. It grouped with the genus Bacterovorax, and it was labeled as SSFD2. These Bacterovorax, like I said earlier, are the non-terrestrial, or let me say they have different characteristics from the Bdellovibrio species. So when we isolated this, it was really strange and interesting to study. But anyway, I didn't include it in my study because of the presence in another genus. This is a summary of all the strains that are isolated. The SKB from soil, the SSB from soil, and their host as well. The SCRB3 from soil as well, using Citrobacter fruendi. The SKB from sewage, using Klebsiella species. And Bacterovorax from sewage, using Salmonella species. All positive for 16S rDNA, only 2 for the host interaction locus. And we use this sequence, the 16S rDNA gene sequence, to construct a phylogenetic tree. We discovered that all the strains, including the Bacterovorax, clustered in the Bdellovibrio group. The SKB clustered separately from the SSB, and it's much more closer to the uncultured Bdellovibrio clone 12L. While the other strain, the SKU and SCR, clustered together, and from the prey range analysis, they also attacked almost the same prey. Interestingly, the Bacterovorax strain, instead of clustering with the Bacterovorax group, clustered with the Bdellovibrio exovorus, which uses another mechanism entirely to attack their prey. But we didn't stop there. We constructed a phylogenetic distance between them to see the variation that we have, taking the threshold that if the distance between them is 0 at 1 decimal place, the sequences are identical. And we discovered that between SKB, SSB, HG100, and strain Tiberius, the distance is about 0.1. While the SKB is about 0.2, showing that the sequence are really close with the uncultured clone of the Bdellovibrio. However, for the SSB, HG100, and Tiberius, they are also really, really close with their sequence giving 0.0. For the SKB, SERB3, same thing was observed. The distance were so close, or the sequence were really identical. But the bacterioviral species, the gap is really wide. It's about 0.7 to 1.0, and this might require a further study. So the conclusion of the first analysis was that Bdellovibrio strains can control, they can serve as potential biocontrol agents because they were able to lyse different groups of organisms, the Enterobacteriaceae, as well as the other groups such as Pseudomonas. And they can also, because they can lyse the Enterobacteriaceae, they can serve as probiotics, just like Atterbury has used Bdellovibrio to determine the improvement in young chicks, and discovered that Bdellovibrio, when the young chicks were infected with Salmonella, the Bdellovibrio was able to control the Salmonella while not affecting the well-being of the chicks. And like I said earlier, the control of Pseudomonas thulasii in mushroom using Bdellovibrio was successful. We also observed that the Bdellovibrio were different. The population of Bdellovibrio were heterogeneous, as earlier described by Djurjevic et al. In 2000, especially based on the heat locus. Some have the heat locus, while some don't have the heat locus. Based on the research on the gram-positive bacteria, it will be interesting to know the protective features or characteristics of Bdellovibrio on gram-positive bacteria. Also, this work can serve as a baseline because of the paucity of emission of Bdellovibrio in Mexico. It can serve as a baseline research for studies on Bdellovibrio in Mexico. The 16SR DNA of the first sequences were deposited in the GenBank. The second study was to carry out the whole genome analysis of the Bdellovibrio strains that we isolated. The two strains that were successful for the whole genome sequencing were the strain SKB and the SSB. The method, briefly, was that the Bdellovibrio strains were cultured with their respective hosts, SKB with Klebsiella, SSB with Salmonella, in Epis buffer. When there was enough lysis, the prey were separated from the predator using centrifugation and filtration using 0.45 micrometer syringe filter. The genomic DNA was extracted using Promega kits and sent for sequencing in my genomics company using Illumina NetSeq sequencing technology. The company sent the reads obtained to us, and we sent them into a pipeline for the cleaning using what is called sickle. We use the sickle to trim the reads to remove the adapters they use during the sequencing technology, or the sequencing method rather. And we did a de novo assembly using spades. We did scaffolding using a software called Medusa. And after, we improved the scaffold we obtained using a software package called Image. Then we were able to get a complete genome for Bdellovibrio SSB, and we were able to get a draft genome for SKB, which contains 20 contigs. The annotation of this genome was done in RAST and was also submitted to NCBI for annotation using PGAP. The downstream analysis that was done was genome comparison, species delineation, using the ANI, average nucleotide identity, and average amino acid identity matrix genome-based distance matrix calculator, and the comparison and visualization of autologous genes cluster using the Autoven program. And in the initial experiment, we discovered that the SSB has heat locus, while SKB doesn't have heat locus. The initial premonition was that the heat locus was missing in the SKB. Therefore, we did a comparison of this heat locus region into two strains to decipher what is actually happening at this region. So this is the report sent to us from MyGenomics that our sequence is good enough for whole genome sequencing. These are the genomic features. We discovered that the genomes of the two strains were very large, as reported earlier by other strains, with SSB giving us one whole genome, and the SKB giving us 20 contigs draft genome. The GC content was high in SSB, while it was low, about 44.8% in SKB. But of major importance is the phage, that the two genomes have one incomplete phage when we analyze using a program called FASTA. The genomic island in the SSB was observed to be 69, while the genomic island was high in the SKB of over 146. The genomic island was predicted using island viewer. The island viewer uses about four methods to predict the genomic islands in a particular genome. The island path demop, the CGHM, the integrated, and the other one, I can't remember now, I'm sorry about that. So, but the successful ones were the island path demop, the CGHM, and the integrated. And they show that, like I said earlier, 69 genomic island and 146 genomic island in SKB. But these genomes were submitted to the Center for Genomic Epidemiology to ascertain if Bdellovibrio is truly pathogenic to humans, if they are considered for application as a biocontrol agent in humans. And the results we obtained said that the input organism, that the genome we submitted to them, shows that the organisms are non-human pathogen for the two strains. And that is showing that promising features of Bdellovibrio as a biocontrol agent. We also try to analyze the different virulence factors in Bdellovibrio, the two genomes that we sequenced. For Bdellovibrio, location of prey is very crucial. Therefore, there were a lot of genes coding for flagellar, I should say, factors, as well as gliding motility factor that help Bdellovibrio to locate its prey when it is on a solid medium or in a place where there is no liquid. The gliding motility factor is what they use to locate their prey. Another virulence factor that we found are the type 4 pillar system factors, which they use for the attachment and invasion of their prey. The other ones were summarized. The chemotaxis factor, which they use to sense the chemical signals produced by their prey. The proteases, the peptidases, which of course was very high when compared with the other strain in HD100. The lipases, the hydrolyses, the esterases, the catalases. But three ones that strike my attention are the collagenase, the hemolysin, and the RTX toxin. Though Bdellovibrio has been described as non-pathogenic, and they have been described to be successful in control of pathogens in humans, yes. But we need to know this collagenase is known for tissue destruction. If Bdellovibrio is housing this collagenase, what is the fate of the cells of human when Bdellovibrio is introduced? As well as the hemolysin and RTS toxin, which are cytolytic in nature, which are pore-forming, which can form pore in cells. But we cannot conclude that these are actually detrimental in Bdellovibrio because they are different. These factors have different biological functions. So it will be important to characterize this collagenase, the type of hemolysins, and the type of RTS toxins to know which type, if they will not be hindering the application of Bdellovibrio in humans. Another analysis we did was the determination of the amino acid identity. It is possible to do delineation of species using amino acid identity. And the threshold is that when the amino acid identity is about 95%, we can group these organisms as the same species. But for Bdellovibrio SKB, the range of the amino acid identity when compared with the other organisms, is between 63.7 to 67.68, which is far, far low than 95%, which is its threshold. But in the SSB, the range is between 66.59 and 94.6, especially with HG100 and Tiberius. The SSB share close amino acid identity with the other strain, the SSB, the HG100 and the Tiberius, showing that this group, the SSB, the HG100 and the Tiberius, could be grouped as the same species, while possibly the SKB could be separated as another species based on the amino acid identity. Another thing to prove is that we retrieve the 16S DNA from each genus used for the amino acid identity. And we try to draw a clustering pattern to confirm the relationship between these strains. And we discovered that the SKB clustered separately, while the SSB, 109G, HG100, and Tiberius are much more closer. Same thing with the EC13 and BER2, they clustered together. This shows the synteny, which further shows that the SKB is much more different from the other strains. There is high synteny between the HG100, SSB, and Tiberius compared to SKB, which is the one below. This was done using progressive Malvey. In short, showing that Bdellovibrio SKB might be a new species, but requires further analysis. For the autologous gene clusters, we considered 6 strains: the W, the 109J, SSB, SKB, HG100, and Tiberius. And we discovered that the 6 strains share 1,036 autologous, autologous genes as a core genome. But the SKB has about 30 unique genes. And these 30 unique genes were submitted to BLAST2Go software to check where they got it from. This is a summary showing the group of the genes, of the unique genes found in the SKB. The larger part of it are hypothetical proteins, while some are insertion sequences, and some are hydrolysis and hydrolytic enzymes needed for the survival of Bdellovibrio. But of one importance is the amylovola biosynthesis protein. This AMF protein is one of the virulent factors in Erwinia carotivora that is a phytopathogen. So we tried to blast the sequences we got in RAST, and we discovered that this particular region found in Bdellovibrio SKB is phage associated. So, brings it to the point that there is tendency of lateral gene transfer in Bdellovibrio SKB. In addition to that, the 13 unique gene clusters were blasted in BLAST2Go software package. And we discovered that these genes have homologue in the in organisms that are different from delta proteobacteria in which Bdellovibrio belongs to. We have some genes that were obtained from the archaea, the spirochetes, the firmicutes, the beta proteobacteria, gamma proteobacteria, Lactobacillus. This has been reported by Gopna et al., and it has been suggested that these are the genes obtained, or these are the sequences obtained through horizontal gene transfer in Bdellovibrio. So, because of the phage-associated Erwinia carotivora protein, as well as the possibility that some sequences were gotten from other bacteria, shows that there is possibility of lateral gene transfer in Bdellovibrio SKB. For the comparison of host interaction locus, we use K-based server to draw a genomic map and went specifically to the region of the host interaction locus to check if there is a presence of this region in SKB. We discovered that it is intact in SSB. The blast analysis of the retrieved sequence at this region shows that it is host interaction locus. Okay? But in SKB, the corresponding region is present, not that it is missing, but there is high variation in the sequence at this region. This is the region for the BD0108 and another region for the BDT0100. When the sequences at this region were blasted, we discovered that they have no heat. When they were blasted the XPASI software. But when they were blasted the NCBI, they had non-significant heat with another protein. Another thing that we noticed was that there is an insertion of another sequence of about 170 base pair, while it is missing in Bdellovibrio SSB. The implication of this is that though the heat locus is present, but there is high variation in the sequences of this heat locus. As you can see, this is BD0108. When the alignment was done using cluster W, this is SKB, there is high variation in the sequence compared to SSB, H100 and Tiberius, where there are conserved region. Same with the region BD0109. We extracted the region from the presentation of this study done by Capines et al., and we used it to align all the sequence, BD0109 sequence, with our own strain. And we discovered that there is conserved region. The implication of this is that BD0108 might be important truly for invasion of Bdellovibrio, but its absence, its variation, doesn't stop predation in Bdellovibrio. However, we postulate that it may affect some features, maybe the prey range or the ferocity of the Bdellovibrio. I'm showing a slide here showing a genome of Bdellovibrio strain, ARSH, obtained by metagenomics. The Bdellovibrio, the BD0108 was completely absent in this strain. But because the metagenomics, they cannot test whether it is predatory or not. But they were able to conclude that it's either this particular strain is housing a different mechanism of attack, or it is an host-independent phenotype. I tried several times to culture the SKB host-independently. I've not been successful yet, but I hope I will get it one day. Conclusion. The genome size of the Bdellovibrio were big, as earlier reported, compared to other obligate parasites. The genome codes for large virulence factors that can aid Bdellovibrio species to be successful as a predator. Though genome analysis in genome analysis of Bdellovibrio, genome analysis as well as the animal model studies showed that Bdellovibrio is non-pathogenic to humans. Promising, yes, it's a good attribute that is really promising. But what about the effect of collagens? What about if the hemolysin can affect humans? What about if the RTX toxin can affect humans? These are open for further studies. So we need to go slow on the application, whereby we need to carry out more analysis on the Bdellovibrio possibility of using it in control by pathogens in humans, and we need to characterize this hemolysin to see that they might not in future affect the application of Bdellovibrio. Because of the phage-associated Erwinia keratomerase AMSF protein and the other 30 unique genes that we acquire from other non-beta bacteria, we can say that the Bdellovibrio SKB has also experienced Asian or recent horizontal gene transfer. The amino acid identity suggests a possible taxonomy review of the Bdellovibrio species. Might not be now, maybe in future when there are more genomes for better comparison. The variation in the BD0108 is not enough. That is, in the heat locus region of BD0108, is not enough to hinder the predatory activity, but may affect some other features like the prey range and the ferocity. Also, the fact that the BD010 region of the Delivibrio is not conserved in all the Delivibrio strains suggests that they might be playing a significant role in the invasion and attachment of Bdellovibrio. Our main hypothesis was to isolate Bdellovibrio strains from the environment in Reynosa and see if they will experience there will be genomic variations. And we were able to isolate strains. And we can say categorically that these strains exhibit genomic variations. Bdellovibrio is a promising antibiotic, antimicrobial agent for the control of pathogens. But considering the influx lateral gene transfer, considering the presence of some virulence factors like collagenase, like RTS toxin, how sustainable can Bdellovibrio be in its application as agent of biocontrol? I would like to appreciate some people for these studies. I would like to appreciate my friend Omar again, Raphael, Litani and Sophia. I would like to appreciate Dr. Eri during the sample collection, and of course, my supervisor, Dr. Mario, for the advice, for the support so far. I would also like to appreciate CODACIT for the grants they give me, and Professor Djukovic in the Hebrew University of Jerusalem for providing the positive strain of the Bdellovibrio AG100 for the positive control analysis. I would also like to appreciate Dr. Brigilio, Dr. Veronica for providing us with some strains during the strain of bacteria for the pre-range analysis. I also appreciate Francisco Hawe for during the analysis using the app software program. And in future, you may visit the doctor and prescribe Bdellovibrio therapy to combat your infection. This was said by Liz Socket in 2008. Muchas gracias for your attention.
Bien, vamos a iniciar con preguntas. ¿Sí? Aquí salió.
Hi, Tayo. How long do you think we have to wait for the Bdellovibrio therapy?
For the therapy.
Yeah.
Anyway, even for drug and for the use of antibiotics, it has to pass through a lot of stages, clinical trials and everything before they can be approved for use. I think the study on Bdellovibrio is still at this stage. Testing in animal models, if they will truly be successful. Yes, they are successful, but there are still some things, some analysis to be done to understand this concept. Is concept, what is going to happen to humans, or are they going to be in future turned against us? We need to find this out. And after that, it has to undergo different clinical trials before. So we cannot say specifically this is when it's going to be available for use.
Do you think it's a long wait or it can come sooner?
I hope so.
Okay.
Okay. ¿Alguna otra, doctora?
Tayo, very nice presentation.
Thank you.
Much better than the last time. Much better. Just one question. When you say that the Bdellovibrio taxonomy has to be reviewed, are you suggesting that you have a new species maybe in one of your isolates or not?
Consider the amino acid identity and some other characteristics. I am suggesting and proposing that it might be a new species, but there is need for further analysis to confirm that.
Is it worth doing finding out if it is a new species? Because probably you need first to find out if it can be used as a therapy agent before doing the other work or not. Or what's first, the identity or the applicability?
The beautiful thing about Bdellovibrio is that they have ability to control. The prey range might be low or the prey range might be high. But the beautiful thing is that they can attack gram-negative bacteria. Okay. What is necessary is that we need to know what is actually attracting Bdellovibrio to a particular spray. If this is solved, then we can target If with SKB, with the little prey range it has, it can attack the Enterobacteriaceae family, which is also a good news. The pathogenic Salmonella, the pathogenic E. coli, the pathogenic organism that we have, the Enterobacteriaceae we have, that means SKB can work. So it is clear that they can serve as a biocontrol agent. There is no doubt about it. But we need to know if they can lose this trait in future. Those are the things we also need to consider. But the fact is, presently, it's working. Okay? So, carrying out other analysis in phylogeny doesn't affect the predatory activity. It can attack.
Okay. Then you would move better for the applicability more than the taxonomy part of the...
That's my interest.
As continuing for this work.
That's my interest.
Okay, thank you.
Ok. ¿Alguna otra? ¿Alguna otra pregunta? Si no, pues le agradecemos al Tayo su presentación. Gracias.